שלום לכולם, ערב טוב, ברוכים הבאים. Sorry, we will have it in English, both seminars, since we have a guest from abroad. So the first lecturer is Lior Zimmerman from Novellus, and you're welcome.
Okay, hello everyone, my name is Lior Zimmerman. I lead the AI research in Novellus, and our company develops solutions for cancer diagnostics and treatment recommendations. A little bit about who we are. We are located in Jerusalem. It's, for people from abroad, is about, I would estimate, 75 kilometers from here. We are about 17 full-time employees. We've raised up until now, in 7 years of operation, we've raised $26 million from OrbiMed, from all the leading VCs that are focused today on healthcare and genetics and pharma. Our clients span a wide range of centers: the MD Anderson Cancer Center, the Fox Chase Cancer Center, and also corporations and companies like LabCorp and Foundation Medicine, for example. And since our primary focus is cancer, this is like the hallmark of our research. I would like to start with like Cancer Biology 101, just to get over the basics, and then we can start talking about the computational research that we're doing in Novellus. So a little bit, I am a bioinformatics scientist. I mainly involved in bioinformatics research. One of the tools that we use in Novellus is deep learning and deep neural networks to investigate the role of mutations in cancer. So, as I said, Cancer Biology 101. So cancer is a multi-stage disease. It develops over years, over decades, and it is caused primarily by DNA damage, mutations in the DNA, and this damage in turn leads to disruption in key cellular processes. Now, just as a really simple example of what I'm talking about, what is a cellular process? So the vast majority of interaction within the cell are mediated by proteins. In our DNA, the DNA that is within our cells is basically a recipe for how to create those proteins. Now, errors in the recipe can lead to malfunctioning proteins. So cells in our body also interact. And for example, when we have an injury, after the injury has finished healing, then special cells in our tissue secrete signals that signal the other types of cells to stop dividing. And in this example here, the interaction is mediated by a small molecule that it penetrates the cell and start a cascade of signals that part of the signal involve interaction between two proteins. So when you have a normal DNA sequence, like the recipe is intact, then these two proteins can interact and propagate the signal downward. When you have an error in the recipe for the proteins, then one of the key blocks in the pathway can't interact with the downward protein, and therefore the entire signal cannot propagate. And so in this example, like if we have a molecule that signals for the cell to stop dividing, then if the signal cannot propagate downward, then the cell cannot stop dividing. Basically, this is one of the hallmarks of cancer is constitutive division of cells. Now, it's important to understand a very important point here. Not all errors in these recipes in the DNA sequence lead to cancer. So you can have mutations. That indeed cause cancer. They interrupt the cellular process. You can have mutations that are completely benign and do not affect any type of interactions between two proteins. And these are actually the majority of mutations. Most of them simply have unknown role in the disease. We simply don't know. Since there are so many possible mutations in all the proteins in our cells, the number of mutations that we simply don't know, we know nothing about, is enormous. Let me give you an example. This is a gene here. This is a plot that have a sequence of a gene as a function of the number of mutations that encountered in cancer patients over the years. So for example, in this gene, you see that position number 600 is enormously more encountered than all the other positions. However, if you count the number of total mutations encountered in this gene, you see that the number of mutations that do not occur in position 600 is not a negligible number, meaning that those mutations that we know about, for example, the 600 one, is, although it's a very big number, but the rest is not negligible. We can't simply let them go and discard them. And so if we take a look at sequencing, so cancer patients, usually part of the diagnosis process for cancer patients is getting tumor cells out of the body using a biopsy, some kind of biopsy method, and then sending those cells for DNA sequencing. That enables us to reveal the collection of mutations that is found within those cells. Now, typically, when we get such a report, it looks like this. You have a list of all the known mutations together with all the FDA-approved treatments and clinical trials and so on. And you have also a list of mutations that are found within those cancer cells for which we know nothing about. We simply don't know if these are disease-causing mutations, like they interrupt key cellular processes, or they're simply benign. Yeah.
Can you not take a biopsy from a regular cell that is not a tumor and just compare the mutation for regular cell with cancer cell and find the differences?
So yes, I mean you need, okay. So in cancer cells, you encounter mutations that you typically see in regular cells, but you need several mutations to work together in order to lead to cancer. So, I mean, one mutation in regular cell can be identical to mutation in cancer cell, but only if you find several of them together with it cancer.
I think this is this question. So this gene here, 600, is this normalized by the number of mutation of this location in non-cancer cells?
No. This was taken from the CBio portal. CBio portal is a data bank for all the tumors that are basically assembled in the database. It's just from tumors, so it's not normalized. So the key point here is that if you take a typical cancer patient, about half to a third of the mutations that are revealed in the tumor have unknown or uncertain role in the disease. And so if you tailor, when a doctor sees this report, basically the tools, the knowledge that he has enables him to tailor the treatment based on a very small subset of the genetic alterations. And that basically can lead to what we typically see in advanced stage cancer, to relapse and resistance. But this approach, where you tailor a treatment just on a small subset of mutations that you're familiar with. And this is the need, this is where we come in as a company. We developed a system that enables us to take this kind of report that I just showed you with a list of unknown mutations and reveal their role in cancer progression or cancer manifestation. So our pipeline is composed of several steps. First of all, we get, as I said, the input to our pipeline is a report, a list of mutations. We synthesize the genes in our lab, and then we transfect cells, a special type of cells that we developed in-house, with those genes. Then we put on key cellular proteins fluorescent tags. These are basically molecules that shine with visible light, for example, green, red, and blue, in response to UV radiation that is radiated on them. Then, for each such gene, we get an image of cells. I'll show you in a couple of slides how it looks. But those images are then fed to an image analysis-based deep neural network that is trained to identify which cells harbor oncogenic mutations and which have benign mutations. And this is basically the output. We send this prediction to the doctors, and they're able to tailor the treatment, in a more informed manner. What we also are able to do, and that, maybe a little bit reaching to the end of the lecture, is able to predict what type of drugs work for what type of mutations, for each mutation. And I'll get to that in a few minutes. So again, as I mentioned, we have a cascade of signal. We tag key cellular proteins with fluorescent tags, and basically have this three RGB image, three color image at the end of the process. So we are mainly interested in two aspects of this problem. As I said, one, prediction of oncogenicity, which mutations can lead to cancer and which not, and prediction of drug responses. Given a mutation, what is the optimal treatment for that particular mutation? So in order to create the pipeline I just described, especially the deep neural network one, we created a training set that is built of 7 genes that are heavily involved in cancer. Actually, no. If you want, I can explain why. So we took a panel of 7 genes to create some kind of a proof of concept. For each of those genes, we took a version of the gene that is known to be oncogenic, a mutation that is known, like these, the bottom row here. These mutations are known to be oncogenic. And we also took a correct version of the gene as like the negative example. Like this is how it should look this is how the cells look when they have a correct version of the gene versus this is an image of the cells when they incorporate a faulty gene.
There's only one faulty per each time?
Yeah.
Only one faulty?
One faulty. Yeah, it's a good question. Whether one type of mutation behaves like all the rest. This is a good question. The answer is no. We have some mutations that behave completely different than the rest. But this is further along. Yeah.
I didn't get why you do your first time the RGB.
Excuse me?
Why do you need the RGB to understand where is the mutation active? The color, the fluorescent that you put on the mutation.
Yeah.
Why do we need it?
It enables us to distinguish between benign mutations and non-benign mutations. Okay? And I'll show you why the network is able to. We've seen it in many examples, but the network is able to distinguish other types of interesting properties. And I'll show you. I'll get to that.
Is the correct version of the gene the same in all people? Because there
These
are
genes,
genetic differences between different people.
Right. So these genes are very conserved. Even if you look at like monkeys, these are very conserved genes that are heavily involved. You have some minor changes, but the function is the same.
So the host cells that we are using don't have an intrinsic version of this gene?
Yes, but
Itself?
yeah, it has an intrinsic version. Actually, the cells we're using are cancer cells. Yeah, they're HeLa cells. But the way you make these images is by overexpressing these proteins. So you have like several orders of magnitudes more the version we inserted versus the the inherent version of the cell. It's like 1,000 more.
So these experiments are from cell lines or from actually cells in people with the tumor?
No, it's from cell lines. The input is just a report, just a list of mutations that enables us to plug in into all. We don't require any biopsies to be sent to us.
You base your decision on I mean, it's a neural network, so maybe you don't know, but does the shape have any meaning, the cell structure at this point? Because if it doesn't, then it's just a three-dimensional signal. The entire image can be summed up as just three-dimensional signal, right?
So yes, I'll talk a little bit about what we think the network look at. What are the important features that the network extracts. But we see that cell shape is definitely a contributor to the decision eventually. Especially, for example, the distribution of color over the cell. Like if you have some mutations where the distribution of the color is like really concentrated in the nucleus, for example, while you see the opposite for other mutations. So it enables, and I'll get to that, but the results part is, we have some results for sure.
The visual analysis is done per cell or per culture? Because if it's a mutation that will cause a huge proliferation of the cells, so eventually you will have a full mutation because of the number of the cells.
Right. So these actually, it could be an important feature. For example, if you have like a really dense cell count, for example. So yeah, I mean, this is we look at it as a culture. Yeah. And in a culture, you also, since cells rarely operate, as one unit, culture is also meaningful. We think that it's meaningful, and I hope I'll be able to convince you. Any more questions?
So if the shape counts, why don't you feed the network with a fourth channel of the raw image, not the...
You mean like segment the cell out and then just train on the cell images?
No, just illuminate the scene, the culture, and take a standard image.
Like a bright field image?
Like
just the shape?
Yeah.
Yeah, so this is actually an interesting point, because if we could do that, then we could save the entire process of like tagging the proteins and just give them an image of the cell. But we see that the distribution of those key cellular proteins is very important for the distinguishing between mutants and wild type. And the location of those proteins, whether it's in the nucleus or not, is very important.
It's sort of a functional tagging. It's like the tagging that you do, it's functional, right?
It's functional. I hope I understood.
Like functional meaning the proteins, the location,
Yeah, I mean, those proteins, when they're in a regular cell, they participate in those interactions, and they move from cellular compartments, as time varies. So when you have in cancer, they are stuck, for example, in the nucleus. So that is one way that the network is able to identify and distinguish between the two classes. So for this purpose, we created a novel architecture that is typically used for, when you get an image of culture cells. There are many studies that describe this kind of a variant of this architecture. What we added On top of that is division to resolutions. So let me go over it like briefly, and then I'll describe a little bit about the rationale behind this architecture. I know that there are a lot of architectures out there. Okay. So the first step here is we take the image of the culture of the cells. We have it in RGB in three color image, and we divide it to three different resolutions. For these three, I hope you can see the numbers: 1024, 512, and 256. Then we partition them to patches. So for the case of the higher resolution, we get 16 patches. The middle resolution, 4 patches, and the lower resolution is just 1 patch. All those patches are fed into a series of convolutions and pooling layers. Eventually, after a series of 5 of convolutions and pooling, they end up, after a global average pooling, they end up as a vector. Each patch ends up as a vector with size of 64. So at the top layer here, all those patches, all those vectors, size 64, are concatenated and create what we call an embedding. The embedding basically represents the image with all those resolutions, all the patches. This is a complete representation of the input. I'll get to that. I'll explain the rationale. Okay, so the embedding is then fed to a fully connected layer, which is connected to one output neuron, which eventually, output 0 or 1, whether it's oncogenic or not. Now, the rationale behind this architecture, I know there are a lot of architectures out there. The rationale here is basically trying to capture local and global features simultaneously. So if you take a convolutional neural network, like, I don't know, just a regular one, for example, just for simplicity's sake, so it is able to capture the local, or if you take a high resolution image, we found that it captures the local features much more than the global one. So for example, for a global feature is like the density of the cells, or the amount of signal, the amount of green color in the image, or stuff like that. So we found that this network actually is able to capture global features and local features. I'm sure that networks like ResNet, which have like 10 million weights, can do just the same. This network actually has just a fraction of the weights. It has 100,000 weights, and some parameters. So it really is fast, and we found that since we are working in patches, it can you can scale to and train really fast. And I hope that the results can convince you later that it works really well. So we talked a little bit about the type of features that are encountered by the network. And we put here a list, like a non-representative lists that basically describe the features we think are interesting. For example, clusters of cells. The more clusters of cells you encounter, you can hypothesize that it corresponds to higher division rates. And you have here some nucleus and time enriched. That basically means that there is a lot of DNA in the cell that also is a potential features, and so on. There is a list here, and I'll show you how these features contribute to predicting other interesting properties of the image. For example, what type of gene is expressed in the cell, not just if it's oncogenic or not, but what gene is expressed. So we took this training set that I described earlier, and we trained our network to predict wild type and mutants. And for most genes, we encountered almost very high accuracy. The ROC here demonstrate you have the numbers for each of the genes. The average is about, since BRAF contains a lot of examples, the average is about 96. The accuracy is also above 93%. So cases, yeah.
The goal is not the detection. The goal is once you have a patch, a small patch with a specific cell, or it's to classify it. It's not doing both the detection and classifying the specific patch, right?
The goal is to take an image of cells, like a culture, and predict whether the cells contain mutant or cancer-causing mutation or benign mutation. That's the goal.
Okay, so you can have several types of this and several types of that, and you just give a global label to the image eventually?
What do you mean, please?
So the previous image, I noticed that there were all kinds of, I guess, subcategories, right?
These are examples of features that the network,
okay, so
perhaps we think, take into account when it predicts mutant or benign.
okay.
Can you test this hypothesis somehow?
So we tried, I mean
this
is
to estimate what are the features that they are network. And another question, how many images do you, in the next slide where you show results, how many
yeah. So to the first question, we analyzed the performance of the network using tools like SHAP or Deep Explainer and so on. And this is just by visual interpretation. This is our interpretation where we saw, for example, when you look at the output of Deep Explainer, you see images of clusters of cells. They all shine up versus where you see them separated, they don't show. So, I mean, it's a visual interpretation. I'm not sure. That's why I said we think. Yeah. And to the second question, so we spent about 2 years creating this data set I just described. It contains about 65,000 images for each of the genes together. So yeah, this is the performance over the training set. So again, we're happy, but the goal here is to predict mutations with unknown role in cancer. And I'll show you that this example show you that we can distinguish between one mutant and one wild type and one correct version. Okay. Another interesting property of the network is the ability to predict what gene is expressed in the cells. I mean, we have 7 of those. Each of them have different features. Is there a difference? Because when I look at those images, I have no idea what type of gene is expressed in those cells. But apparently, the network has a pretty good ability to distinguish between those what gene is expressed in the cell, and it can even distinguish between the one reporter. These 2 have a different reporter, and these 5 have a different reporter. So it can really distinguish what type of gene is in the cell, which is very interesting because some cancers have several genes that are involved in the disease, so it's useful for it. Although it's in our roadmap, we haven't got to several genes together, just one gene at a time. Now, as I mentioned, we're also interested in seeing how mutations with unknown role in cancer behave. So for this purpose, just to see how it looks, we took the embedding created by concatenating all the representations of the patches, and we did like a dimensionality reduction analysis. And that really shows that if we take, for example, mutations in a gene called KRAS, we have like a population here that is concentrated around a known cancer-causing mutation, the one that we trained on, while you have some KRAS variants here, which is probably not part of the cancer-causing mutations. Same for the other types of genes. You have like a cluster of known cancer-causing mutations, well, you have a distribution that is mostly, if you see here, for example, wild type, the word wild type here, if a mutation is closer to a wild type, then you can hypothesize.
What of the axis here?
So this is a principal component analysis. So the x-axis is the first principal component, and the y-axis is the second.
Which is the same for all genes.
So a word about principal component analysis is basically a way to take a multidimensional vector and reduce it to 2 dimensions, by maximizing the variance between the same. Okay, so so this is the network, the way we trained it. How are we using it today in Novellus? So one way, and this is an image that was taken from a paper that was submitted recently to the Nature Biotechnology. So one way we can use it, as you can probably guess, is taking a gene and collecting all the mutations that are encountered in cancer patients over time and annotating them, basically giving the service to the scientific community and saying, like, look, we have the c-KIT gene, which is frequently mutated in a lot of cancers, and we annotated all the mutations you have ever encountered. We took a panel of about 200. And so you see all the blue here, all the blue lollipops here are mutations that, the ones that are both, let's say, 0.5, are oncogenic mutations that are not familiar to the scientific community. There are quite a few here. So these are novel mutations that we discovered using our platform.
But we're only using images from people who are known to have cancer.
We are using mutations from people who are known to have cancer.
So you have no idea if you take 10,000 images from people who are considered healthy, maybe you find the same mutation there and no cancer.
Yeah.
You have no control. You have no control.
Right. But the key here is that you rarely get mutations in cancer. Cancer is rarely caused by one mutation. You have cancer-causing mutations in many types of cells.
Even stronger need for control, because you might have 6 genes mutated here, and then you might have 6 or 5 genes, the same mutation in other people, and you don't know who is the odd gene out or whatever.
Yeah, you're right. I think the rationale here is basically designing a treatment plan. So when you have, for example, you get 8 mutations, right? And you see here that 6 of 8 of those mutations are cancer-causing mutations. So what we offer here is a way to tailor the treatment to maximize the
what
mutations.
You have here is an hypothesis saying that genes and genes alone cause this cancer. You are only looking at a certain subset and you have no control. So your hypothesis is a work or not, but since you have no control, you cannot prove it.
But how would you imagine this type?
If you take a series of cells from healthy people, considered healthy people, and you show that only in people having cancer, only in them do you have this convalescence of mutated genes, and healthy people, this rarely ever happens, and 0.1% of healthy people only have this. Then you have some scientific backing for your hypothesis. What you have here is I take cells from people who are not healthy, and I find these findings. But are these findings the reason they are not healthy, or are they not healthy? And these are some of the things that I find.
I'll be happy. I mean, I have an answer, but I'll be happy to continue this time. I think there is a part which is correct, but a little bit that is impractical for this type of thing. Yeah.
So, is the importance of the features we learned from known mutations actually imply that unknown mutations are relevant? So, we've learned images with known mutations, right? In the training set, you knew what mutations are represented in that training set, and the network learned a set of features to predict the kind of mutations that exist. Right? So there is also a representation vector that hopefully lives in the space of mutations that we can use to separate between mutations that are cancerous versus everything else. So what I'm asking is the features that we learn relevant themselves to the endpoint that you're looking for.
So if I understand the question correctly, so there are mutations that behave different than the mutations we have in the training set. I mean, there are even if you look at the same genes, some mutations have different features, and we see that in our results. They simply appear as false positives, for example, or this type of things. I mean, you see them like get a score that corresponds to, for example, benign mutations. Yeah, we see that, and the goal in our long-term vision is to incorporate more genes into the system, so we'll be able to provide like a better prediction. But this also forces us to change the biological assay, because some mutations require different settings and so on. They behave differently and require different tagging.
We have like 5 minutes.
Yeah, I'm getting to the end. Okay, so the last thing I want to show you is how we predict, how we basically tailor a treatment to a mutation, how we decide which treatment works in the optimal way per mutation. The way we do that is we take a population of cells that are incubated in this plate, for example, and we incubate them with increasing or different concentrations of drugs, cancer drugs. And then we take the fluorescent microscopy image of each of those population of cells, and we feed these images to the deep neural network, and we get this type of plot here that the x-axis is the concentration of the drug, and the y-axis is the output of the network, where 1 is highly oncogenic mutation and 0 is like benign or correct. And we have this example here shows 2 types of drugs and 4 types of genetic alterations of a certain gene called c-KIT. So, for example, in this alteration, based on the prediction we provided, the dasatinib works much better than sorafenib, and it's also basically backed by literature, which is the optimal treatment. You have dasatinib works much better in lower concentrations. This type of alteration also shows that dasatinib works much better. And another thing that we can see that the wild type, which is the correct version of the gene, is unaffected by the drug, which is desirable property. One thing that is interesting and alludes to a part that I won't get to that, but is described in the Nature Biotech paper, is that we discovered a natural effect of the drug. We found a paradoxical activation of the pathway that is been demonstrated by the increase of the signal here. You see that for dasatinib, for the drug dasatinib, you see that the signal is increased in concentration of about 500 nanomolars. So this basically means that the drug dasatinib may lead to increased activation of the pathway, which means that it could be less optimal in higher concentrations. If you give some patient, for example, a large amount of the drug, then you may cause exacerbation of the disease. And this is also something that has some evidence in the literature. That's it. I think I don't have time to get into our clinical work. This is the clinical work we've done with the Institute of Paris. But thank you very much. And if you have any questions.
Maybe you can say a word about like comparing. Like I'm not from the field. Maybe you can like give reference what is being done in this field. What are other techniques being
Best
used?
practice in the field.
Like what is the state of the art when
So if you look at this task, you can look at it from two different perspectives, from the experimental perspective and from the computational perspective. So there are a lot of works, a lot of studies that involve, like, let's predict the effect of mutation, but just from a computational standpoint. Let's look at the structures of proteins, how they change, and the change in the folding energy and so on. And you have like experimental methods that from taking a population of cells from the tumor and testing them with drugs and so on. So I mean, it's a whole world out there, and just saying. And I think that our method is the only one that I'm familiar with that takes both computational and the experimental method and combine them together.
So, but taking this sort of signal compared to, for example, taking an NMR signal or I don't know, other physical signals, isn't like is most of the information there? Are you sure that you will be able to do this operation? And I don't know.
So sure is a strong word when you come to cancer. I mean,
Yeah.
We have some genes that we have high confidence for, the robustness of the network and the entire platform. Some genes work to a much less degree. If you compare it, for example, for a CT scan or NMR, any type of method like that, then, I mean, CT can predict drug responses, at least not that I'm familiar with.
Yeah, I'm talking about the cell level, not CT.
So
for
the-
example, NMR signal at the cell level. So would that be? I just wonder.
Yeah, that's a good question. I mean, whether you can predict the type of mutation a patient has just by looking at the CT scan or the NMR. That's a good question. I've seen a study that predicted whether a patient has EGFR mutation or not just by looking at the CT scan. So I think there is a basis to it, but I'm not sure that it can be as accurate as this kind of method. Like, can you pinpoint the exact position or mutation? I'm not sure. I don't think so.
Can you say a few words on business implications of this technology? What's the primary customers? Is it pharma? Is it clinical treatment?
So we have like two avenues where we work with businesses. One avenue is working with diagnostic company like Foundation Medicine, LabCorp. They offer their clients DNA sequencing of tumors, like the report that I showed at the beginning, where with a notation for the doctors, which mutation has clinical implication, which not. This is one avenue. Another avenue is coming to drug companies and saying something like, yeah, we have a system. You have several candidates for molecules. Give us those molecules and we can tell you what patients you should include in your clinical trial to maximize the efficiency of the clinical trial, the success rates, for example. Because one of the things that those companies usually encounter that they want to include as much patients as possible, but they end up having really bad, most patients die eventually. So you can maximize the success rate of the clinical trial if you include only ones for which you get informed decision.
I think that if you would use your system also to try and assess different drugs on the same mutation, so another potential client could be pharmaceutical drug companies that if, for example, you say drug X is the best for this kind of mutation, so this information is very valuable for the pharma company to know that doctors may recommend for each patient that has this mutation that this specific drug will be the best to use.
Yeah, you're talking about adding indication on top of existing drugs. Yeah, that's also interesting. We work with that. And actually, recently we've pivoted to drug development. We kind of have a really negligible amount in the diagnostic space, and we moved to drug development. yeah, the big money is in drug development. Okay. Any other questions? Thank you very much.
We will take a few minutes break and set up the next speaker and enjoy, and we'll call you back in a few minutes. Thank you.
Elias.
תודה רבה. מאוד מעניין. זכותי, אני לא מעצבת את זה. אני, כן, יש קטע של ביולוגיה וגנטיקה, אני יודעת,
אני מקווה שהפכתי את זה לברור.
כן, דיברנו מאוד יפה.
נושא מורכב.
המחשב שלך, זה שלנו?
לגבי השאלה שלך, אני בטח, אתה צודק. יש בזה...
אתה רוצה להרצות על המחשב שלנו? על הנעים גם איך להציג אותך?
איזה שני אנשים פה בחלק משווה את הגנטיקה שלהם. קודם כל 99
רגע, אני מישהו מחבר אותך כי אני רוצה לעשות לך את זה.
כן, העניין הוא שכשיש לך מוטציות, יש לך קודם כל 500 גנים שאפשר לבדוק שהם מעורבים בסדר. כל אחד מהם יכולה להיות מוטציה.
אתה חושב שהם מעורבים בסדר. אתה לא יודע.
יש עדויות ניסוי. ו-500 גנים שיש עבורם עדות, כלומר במעבדה, שעבורם
מההיכרות שלי, ואתה לדבר על כמה עשרות אונקולוגים לאורך השנים, לא באמת מבינים ביולוגיה של סרטן גם היום. זאת אומרת, יש היום מעט מאוד סוגי סרטן, סוגי סרטן איפה לפצח במדינות ביולוגיה, ואז יש להם תרופות ביולוגיות וזה עובד נהדר. עובד הרבה יותר נהדר ממה שעבד קודם, ואז כל האחרים שאתה שם את הכימותרפיה, אתה בעצם מפציץ את הגוף בצבען שנועד להרוג תאים צעירים שהם יותר פגיעים, ואתה מאמין שהוא ירוג את הפגיעים יותר ממה שהוא ירוג כל השאר, ולפעמים זה עובד.
רק נקודה קטנה, השלכות שמדובר עליהן פה, תרופות שהן טרגט יותר.
זה מתן שאנחנו בעצם את הדבר הזה, אני להגיד שלא מבינים את הביולוגיה. לא מבינים. ולא מבינים למה אצל נשים מסוימים קורה להם 30, אצל 90 ואחרים יכולים להשם כל החיים ולא קורה להם כלום. יפה. ואז אתה בא ואתה אומר, אוקיי, לקחת אוכלוסייה מסוימת, יש לך שזה בגנים. ולא נניח באורח החיים, ולא נניח בסביבה שאתה נמצא בה, ולא נניח כל מיני דברים אחרים חיצוניים.
כן, יש מה שהסביבה, זה בסופו של דבר גורם זה ההשפעות.
יכול להיות במצבים, כמו למשל בעישון שנוצר מעבר לזה, זה נוצר משקעים. למשל, יש בריאות, נוצרים משקעים, כלום שמסתבר שלא עובד אותך. אז אני אומר, יש גם הוכחות קליניות שמראות שהסביבה שנמצא בה, בלי קשר לשינוי זה, יוצרת אצלך מפגעים בגוף. כל איזה ככה. בלי קונטרול, איך אתה בודק פיתופיזם?
אז מה, אני יכול להגיד לך שכל מה שאתה מדבר עליו, בסופו של דבר מדובר גם אם יש לך כל זה מדובר באוסף של מה שאתה אומר, יכול להיות מוטציות, שאותן מוטציות, סרטן לב,
אבל לא קורה כאילו.
העניין הוא שזה אינטראקציה כל כך מעצבים, כל הגנים בגוף, בסופו של דבר, מניפסט אצלו. עכשיו, אם יש דרך טובה למדל את כל האינטראקציות האלה, המטרה שלנו היא אחרת, אנחנו באים מזווית הרבה בפרקטיקה. יש לך מוטציה, המוטציה הזאת ידועה כגורם מצב. למה ידועה? בגלל שהיא מוכיחה,
99%,
לא מוכיחה, כן, גורם מצב, איך מראים את זה? יש דרכים ניסויות בתאים להראות שמוטציה גורם מצב. זאת אומרת שהיא אינטראפט קיסר עבור האוסף של מוטציות כאלה שכל אחת מהן אינטראפט קיסר עבור. עכשיו אתה יכול להגיד, אנחנו באים מזה בכלל, שאומרים, אוקיי, אני יודע שיש לי פה 5 מוטציות שעבורם הרשת מזהה, בואו אני אנסה לחצות כמה שיותר כאלה. עכשיו יכול להיות שיש מוטציות כאלה בתאים בריאים, זה לא אומר שהם לא גורמים לסרטן. זה אומר שעוד לא צברת מספיק מוטציות כדי להגיע לסרטן.
תודה רבה.
לא.
אגב, יש עבודה שפורסם שדווקא בליאור דווקא נכון על
גיל של האם אתם וכן זה עכשיו אם חשפת את הגידול באופציה כן זה כאן נכון עכשיו יש לנו מה טרנזישן יש קומוניקציה ואלה החידושים שמתיחות כתרופות שמיועדות שמכוונות המקרים בחולים שהם כתיבים להם לא בזמן והתכלס שאלה אם
אנחנו עובדים על אספקט מאוד צר. אתה מתקצר, זה אומר, קינזים, זה מתקצר. יש את האחרים. זאת אחת האופציות אנחנו לא לוקחים בחשבון את ההסתברות ההיפותזה שעומדת מאחורי decision medicine בכל מצב they are varying to עכשיו זה לאו דווקא נכון, זה לאו דווקא נכון, יש מולקולות שעברות יותר וגדולות יותר כרגע תראה, העניין הוא שיש פה יש פה פשוט יש היפותזה פשוט שברוכחה בכמה מקרים שיכולה לטפל בהרבה סוגים של גידולים בעזרת שיש להם את אותה מוטציה בעזרת אותה והראו את זה בכמה
because of the genes sometimes it's
למשל בלי לאפשר כביש משמעותי
wait, maybe it's
רואים אותה גם בכל אז זה בעצם יש פה כמה מקרים שבו מקרים כאלה מקרים שהגידולים יש להם אוריינטציות להמליץ לרופא יש כבר טיפול במלנומה, אבל קודם כל תמיד מדברים פה מי עושים second line, third line of treatment אבל בסדר אבל זה חולים שכבר לא הגיבו לכלום ואומרים אוקיי, מה אנחנו יכולים לעשות בשבילם הרבה פעמים
you're clicking. So the last line of defense. What time? Now it's next week at Chip. But it doesn't appear either. This one? No, it does. In the PowerPoint itself, does it appear? And if you click it, then does it work? I think I said something like
הנה. אתה רוצה לנסות פה?
control panel. The classical view. Says in classical view display icon. Maybe view by small icon. Maybe settings tab advanced. Something like that. It's hardware acceleration. It says. Then it should be. Then it says advanced troubleshoot or something, and then advanced scan. No, it's probably advanced settings. Yeah, and then it said something like, well, the advanced settings are not here. Click on the troubleshoot. There's troubleshooting. Otherwise, it said disable hardware acceleration completely by dragging the slider to the extreme left. Maybe try here. Hardware.
You needed us to speak in English? It was in Hebrew or in English to talk? I don't remember.
It was in English.
Okay, I don't know.
It was for me.
It was okay. Okay,
like,
good.
it's super nice.
Do you want to give me a discount key and I'll try in the meantime?
Yeah, it's probably easier. To do this.
Let's do this. Okay. Yeah. Do you have the... I'll plug this one in, yeah? I need an adapter.
It's for the pointer.
This is for the pointer. So I need an adapter here and I don't have, no. Is there one? I need an adapter. I'll go get one. יש לך דיסק און קי? במייל? כן. לא בהכרח אני יכולה במייל. אתה רוצה לשלוח רגע ל... אתה רוצה לשלוח במייל רגע לכן? או אליי? לא.
זה האוכל של חן?
כן, נראה לי. אתה רוצה לשלוח לכן רגע? מה? מעולה. אחר כך זה לא זה.
וצריך לשתף גם בהנגאוטס.
אנחנו בהנגאוט משותפים.
אוקיי.
כי הפגישה פתוחה.
אליאס התחבר כאילו?
רואה? פה.
לא, אנחנו צריכים שהפרופיל של אליאס שתהיה תמונה.
זה המחשב שלנו, אנחנו מרצה מהמחשב שלנו.
הוא לא מרצה מהמחשב שלו בסוף?
לא, משהו עם ה-GPU והמסך, תמיד עושה לי בעיה. חלק מהמסכים זה כאילו לא אומר...
הוא לא מחליף את
סרטון פשוט מתקבל על התמונה.
הבנתי, כן. טוב, זה יותר בטוח.
זה אתה? לא, זה לא אתה.
אז שמתי עכשיו.
אני לא רואה את הדיסק שלך. ליאור זה?
תושיבה
לא,
משהו?
זה לא אתה.
תושיבה.
אני לא מצאת אותו.
תושיבה. It's planning for dance.
רואים אותו?
אוקיי.
PPTX, אנחנו יכולים לקרוא PPTX? כאילו
חושב שלא?
תוריד, אפשר לעשות, יש פה את הפייג'ס שלנו.
יש לה... אני מציע לה.
איך נקרא? keynote או pages. יש פה.
מעולה. נראה לי שכדאי שנתחיל.
רק פה זה בלוסט שווה לה כאילו.
זה לא
צריך לעשות לו
מחוברים?
שיתוף בדיספליי.